Good evening, everyone. Myself, Dr. P. Samuel, Assistant Professor of Biotechnology of Ayya, Nadar, Janaki, Ammal College, Sivakasi. Through this video tutorial, we are going to see about plasmid DNA isolation. Right? So let us move into the topic. Fine. The below illustration is a bacterial cell. You all know that it is a prokaryotic organism, right? Prokaryotic organism means a very primitive cell, which means they are underdeveloped in nature, right? They are very primitive. Their cellular architecture is also very primitive, and this bacterial cell is very unique in its behavior because it has a tight or a defined cell wall, which is the outermost barrier, and it also protects the bacteria from external factors. Added very next layer to the cell wall is plasma membrane, which encloses the cytoplasm and other cytoplasmic contents. So while going into the core of the cytoplasm, we could see two distinct type of DNA molecule. One is called the chromosomal DNA, another one is called the plasmid DNA, right? Fine. See, the space between the cell wall and the cell membrane, the space between the cell wall and the cell membrane is called periplasmic space, right? Periplasmic space. And we'll move on to the next slide. See, in terms of plasmids, we can classify them into two different types. One is called the stringent plasmid, another one is called relaxed plasmid. So you may ask questions, sir, what is a stringent plasmid? Can you please define? Right? What is a stringent plasmid? Actually, the term stringent refers to the existence of small number of copies of plasmid DNA within the bacterial cell. Say, for example, in case of the stringent type of plasmid, we could notice around 1 to 10 copies of plasmid inside the cell, not more than that, right? As we all know that plasmids are extra chromosomal DNA, they replicates on its own, right? Everything goes on, but the replication as well as the number of copies exist within the cytoplasm is 1 to 10 copies. If the number is limited, or if the number is less than 10 copies per cell, then that kind of plasmid is said to be stringent plasmid. Then what is relaxed plasmid? Another classification. What is called a relaxed plasmid? So as the name suggests, their copy number is more. For example, greater than 10, less than 100. So this is what the characteristic feature of relaxed type of plasmids. Their copy number is greater than 10 but less than 100, which means what? The copy number of relaxed plasmid is more when compared to the existence of stringent plasmids, right? Okay. We'll move on to the next slide. See, in molecular biology, the role of plasmid is very important because it is being called as the cloning vector. It is called as cloning vector. What is a cloning vector? Means these plasmids can be used to carry the exogenous genetic material into the bacterial cell, right, for the production of recombinant proteins. So in such cases, it is necessary for the researcher to isolate the intact plasmid for further manipulation. As you all know that plasmids are only present in prokaryotic cell. It can also present in eukaryotic cell, but that is not a natural process, a process called transfection, through which we can transfer the plasmid DNA into the eukaryotic cell. Right? Natural existence of plasmid in eukaryotic cell is not yet reported. So the ultimate source of plasmid is the bacteria, right? So to initiate the process, right, to begin with the process of isolation, first what we need is the culture in more numbers. I mean the bacterial culture in more number. So for that we are going to grow the cells in a specialized medium called LB medium. What is LB medium? Lurea bertani agar, right? Lurea bertani agar. So since it is a process to grow the cells in more number, we are preparing LB medium in the form of liquid form, that is called LB broth. LB broth. LB broth refers to Lurea bertani broth. Lurea bertani broth. So Lurea bertani broth is recommended for growing cells, particularly for the DNA based studies, right? If you are going for isolation of DNA or for quantification of DNA, so for that purpose, or as in a simple way, for growing cells, particularly for DNA based studies, we can grow them in LB agar medium, right? So after the growth, I mean after a particular time period, which means after 24 hours of incubation, right, a small volume of cell, which means the broth containing the cells, are transferred into a fresh tube with the help of a microliter pipette, right? Exactly 1.5 ml of bacterial culture is transferred into vial. The volume of the vial is 1.5 ml. 1.5 ml. This is otherwise called as Eppend of tube. EPPENDORF. Eppend of tube. Right? So after 24 hours. So what is happening during this time means, since it is a nutrient medium, since LB broth is a nutrient medium, cells growing in the media, particularly the E. coli, the model organism in molecular biology, growing in the LB broth, utilize the nutrients present in the media and grow richly, right? And after this particular time period, after 24 hours, the culture is transferred into a fresh vial. Fine. See, this is how you have to transfer the broth containing cells into a fresh tube. Now the tube is filled with broth. The broth in turn contain number of cells, right? Contain number of cells. Fine. After this process, the next step is centrifugation. So centrifugation is a process of separating high density molecules based upon their sedimentation coefficient, right? Sedimentation coefficient, right? So this is the principle of centrifugation process, right? It is a machine. Centrifuge is a machine. Centrifugation is an experimental procedure which enable us to separate high density substances from low density substances, right? Fine. So this centrifugation process is carried out in a restricted condition, which means the centrifugation process is done at 6000 gram for 15 minutes. So which will enable or which will separate the high density cells at the bottom, right? High density cell at the bottom. This is called pellet. Pellet usually contain high density molecular weight substances, and the upper layer is called supernatant, which usually contain low density substances, right? So after centrifugation process, this will takes place, right? Next, and following the centrifugation process, it is our duty to discard the supernatant, right? So what is supernatant? You all know that the layer containing low density molecules, right? So that is discarded, which means what? That is removed from the vial, right? No supernatant here, right? So following discarding the broth, I mean the layer with low density substances, the pellet alone separated. Pellet purely contain the bacteria. Pellet purely contain the bacterial cells. And in the next step, here we are going to subject the cells for plasmid DNA isolation. So it can be done with commercially available kit, DNA isolation kit, which is supplied with different chemical reagents, which in turn facilitate the isolation of plasmid DNA from the bacterial cells. Fine. And in this step, we are going to add the reagents. So to the pellet, what you are adding, what we are going to add is the suspension buffer. What is called a suspension buffer, and what are all its composition? See, it contains tris EDTA, right, which is used by the researcher to destabilize the cell wall, because as I told you, cell wall is the barrier of bacteria. It is very tough to break the cell wall of bacterium. It is a barrier, it is outermost protective layer. So in order to isolate the plasmid DNA from the cell, it is our foremost responsibility to break the cell wall without damaging the intracellular contents, right? That is to be kept in mind, right? It is our bounded responsibility to break the cell wall without causing any damage to the intracellular structures. So for that, we are treating the cells with EDTA called ethylene diamine tetraacetic acid, which is a chelating agent. This will destabilize the cell wall. And you are adding another chemical called glucose. So glucose prevent the cells from bursting, right, by osmosis. It may also cause some irreversible damage to the cell. To prevent that, we are treating the cell suspension with glucose. So last, we are going to add an enzyme called RNase. It is to degrade the cellular RNA during cell lysis, because what we ultimately need is the plasmid DNA. Apart from plasmid DNA, all other macromolecules are considered as contaminants here. So we are treating the cells subsequently with different chemicals, which enable us to isolate plasmid DNA in its purest form, right? Next slide. This process is called, this step is called celllysis. Here, it is carried out by a process called alkaline solution, right? Alkaline solution containing sodium hydroxide and sodium diodecyl sulfate. So sodium hydroxide is an alkali, you know that it is an alkali, and sodium diodecyl sulfate is a detergent, right? Anionic detergent. Anionic detergent. Fine. And followed by cell lysis, there is another step called neutralization procedure. So what is neutralization procedure? Here we are going to add a chemical called potassium acetate, which will neutralize the suspension, as well as it will enable us to separate the plasmid DNA from genomic DNA, because genomic DNA is high molecular weight substance, right? So once the genomic DNA is degraded, there are possibilities for the release of proteins into the cell suspension, which in turn contaminate your plasmid DNA. To avoid that, we are going to neutralize the suspension by the addition of potassium acetate, right? Finally, the step called precipitation, and this step enables us to precipitate the plasmid DNA, which will settle at the bottom of the centrifuge tube. See, upon every step we have to continue the process called centrifugation. The centrifugation process alone help us to separate the plasmid DNA from the bacterial cell, and this plasmid DNA is in turn utilized by the researchers, right, to manipulate it, and it can also be used for carrying exogenous genetic material into the bacterial cell for further research studies, right? So this is how you have to isolate the plasmid DNA molecule from the bacterial cells. Thanks for watching the video. I am sure we will meet in another session with an excellent presentation. Thanks for watching. Stay connected with me. Bye bye.